Hi, everyone. Thanks for joining. We'll just give it 1 minute so that Zoom allows everyone to come into the room, and then we'll go ahead and get started. All right. So it seems like there's quite a few people in the room, so let's go ahead and get started. Hello, everyone. Thank you so much for joining our very first webinar of the year. So today we have Dr. Simone Sidoli with us, who will be giving an overview on single-cell proteomics and some considerations for experimental design. Before I pass it off to Dr. Sidoli, though, I'm going to do a brief introduction on Protein Tech and who we are as a company. So, Protein Tech is a leading provider of life science reagents. The company was founded back in 2001, and at that time, it was primarily a supplier of antibodies. And the vision of the company was to be able to provide an antibody for every protein in the human proteome. And that continues to be our vision, and we have about two-thirds of the human proteome covered. In addition to our expansive portfolio of antibodies, we also offer immunoassays such as ELISA kits and IHC kits, and we also have cell separation kits and antibody labeling kits. Within our Protein Tech brand, we also have our humankind branded cytokines and growth factors, and all our proteins are expressed in human HEC293 cells, so you can be confident that they have the native folding, post-translational modifications, and glycosylation that you would expect to see in human proteins, and that ultimately increases their bioactivity. We also have our Chromatech branded nanobody based reagents. So, nanobodies are about 10 times smaller than traditional antibodies, and they can bind to their targets with really high affinity, making them ideal for certain applications such as immunoprecipitation, super resolution microscopy, and protein purification. And then lastly, we also have a subsidiary of Protein Tech known as Protein Tech Genomics, and they offer antibodies that are conjugated to oligonucleotides that integrate with single cell RNA-seq workflows to analyze a broad range of proteins, including cell surface, intracellular, and transcription related proteins alongside gene expression. So, we are a company that is driven by scientists, and we're dedicated to supporting our researchers and the scientific community through award winning products, full workflow solutions, resources, and continuous innovations. What truly sets us apart is that we are in control of both the supply and performance of our products through in house manufacturing, which allows us to offer the largest portfolio of antibodies covering the human proteome. To make testing easier and more convenient, most of our antibodies are also offered in 20 microliter trial sizes. We also put an emphasis on rigorous product validation, and we provide open access validation data for transparency and reproducibility. Whenever we are testing new products, we validate with relevant sample types, appropriate experimental conditions, and using knockout knockdown models whenever possible. Also, as antibody experts, we have in depth knowledge of immunoassay applications, and we can offer outstanding technical support for our customers. We also stay on top with cutting edge technology, such as our proprietary antigen specific B cell cloning and engineering platform, which we use for the development of our recombinant antibodies. And finally, we are a global company and we have 7 inventory sites and manufacturing and research sites on 3 continents. So we are able to offer next day delivery to many areas in the world. So, as a company, we really do measure our success by our customers' scientific success, specifically by helping them publish impactful research faster. And so, this graph here on the left shows that over our company history, our products are being featured in more and more publications each year. And as of now, we have over 400,000 publication citations, making us one of the top 4 cited antibody companies and also one of the fastest growing companies in product citations. And then just briefly, I wanted to share some of the latest initiatives that we've introduced on our website last year. The first is our focus on epitope mapping. So, using experimental and AI approaches, we've actually started mapping antibody binding sites back onto the 3D protein structures to offer more transparency about our products to our customers. We believe that by getting epitope level insight, researchers can make better decisions on the antibodies that they select for their experiments and predict whether an antibody might perform for certain applications. We also introduced an immunogen selection function so that researchers can actually filter for antibodies that are specifically generated against their region of interest. Again, this was implemented in the hopes that we're offering, more transparency and to make reagent selection easier for our customers. And then lastly, I just want to highlight that we have built off the information from our 3D epitope mapper and database, and we've recently launched an AI-powered search and resource tool on our website called Able. So Able allows you to directly chat and ask for product recommendations, help with experimental design, and access relevant validation data, citations, and resources. So for example, you can ask Able, what products or resources do you have available for single cell analysis? And Able will be able to suggest the best products for your experimental needs so you don't have to waste time searching. So this AI-powered tool is the first of its kind, and it was designed by our scientists with our customers in mind. We're also in complete control of the information that Able learns from, so you don't need to worry about any responses being polluted by outside sources. So if you do have a moment, please do check it out and ask Able a question. You can just scan the QR code right here, and it'll take you directly to Able, or you can always access it on our homepage on ptglab.com. So with that, I would like to introduce our speaker for today, Dr. Simone Sidoli. So Dr. Sidoli is an associate professor in the Department of Biochemistry and Genetics at the Albert Einstein College of Medicine. He completed his master at the University of Parma, his PhD at the University of Southern Denmark, and his postdoc at the University of Pennsylvania. He joined Einstein in 2019 to lead a research program in proteomics, chromatin biology, and aging research. He has received generous funding from the NIH, the Evolution Foundation, the European Union, and the Japan Agency for Medical Research and Development. His lab also works closely with private companies such as Merck, Relay Therapeutics, and CellVivo. So with that, I will go ahead and pass it off to you, Simone.
Thank you, Frida. Hello, everybody from all around the world. Like it's really overwhelming to see how many people are connected, but I hope I can make the next 45 minutes interesting for you. And thank you, Protein Tech, of course, for inviting me to give you an overview about emerging technologies in proteomics. So let me start to share my screen, and I'll start digging right in it. Perfect. So today I'll pick up on the very ambitious task of giving you an overview of single-cell proteomics, like a bit of the history, how it started. This is the agenda of the things that we're going to discuss today. So obviously, I don't mean with this to claim that I'm the world expert of single-cell proteomics. There are many scientists that every day, as we speak, doing like great progress with the technology. But I hope that with the conversation that we're going to have today, we'll bring everybody up to speed about where the innovation is today and what has been done, and what mistakes do not repeat, and what are the venues for to do more. Very briefly, of course, what is single-cell proteomics? Because I'm sure most of you already know. Well, basically, it starts from the logic that when we study a cell population, we don't stop at the information of what the average cell population represents, but we actually dissect individual cells and what the individual cell populations heterogeneity contains. And there is always this example of the fruit and the smoothies, which is maybe oversimplified, but it kind of gives the idea about how much information we lose when we want to identify, for example, a fruit basket based on the smoothie, because there are many heterogeneous things like that compose this smoothie. And that's, if you think about it, that's how we look at most of our samples. But the thing is that even the most simple cell population is heterogeneous. Like you can take a plate where cells all have the same genome, they're all plated at the same time. They can be like in different stages of the cell cycle, which already makes them different from one sort or for one reason or another. And then, of course, if we look at anomalous conditions, then this heterogeneity like skyrockets. As you, for example, one of the most complex aspects of studying aging biology is not only that old cells are different from the new ones, but old cells are in a population that is more heterogeneous compared to young cell. Part of it is because of DNA stochastic mutations that accumulate differently cell by cell, but there are also many other reasons. And I'm not even going to get into the details of tumors because, of course, we all know that in a tumor there are cells that have different mutations, have different epigenomes, they have different phenotypes, and actually this is one of the reasons why treatments for cancers they're not always effective. So basically, not every cell has the same pathological state, and not every pathological state is the same, and not every cell responds to drugs the same way, right? So that's why, being able to understand the heterogeneity of a cell population actually, is the next frontier of science. The next frontier of science, but we're already into that, and of course a lot of progress has already been made. For example, if we think about single cell sequencing, this one is an example of a transcriptomic data set. You can actually amplify RNAs from individual cells with PCR. You can attach barcodes, so you can multiplex the analysis for thousands and thousands of cells, and then once you plot the distribution, and we actually will go through a little bit of that analysis as well in this presentation. If you plot the distribution of cells based on their RNA population, so for example, all the cells that have the same transcriptomes, they're clustered right next to each other in this kind of graph, you can start to divide the cell population by cell type. And actually, if you have biomarkers within those clusters, so you actually know that this specific group belongs to a specific cell type, you can actually start to infer, what kind of cell types are in there, and also how heterogeneous are these cell type population. You can see that some of these cell populations create much bigger clouds compared to others that instead are much more uniform in terms of RNA expression. But, of course, we are interested in the proteome level for many reasons. One, if anything, because proteins are almost always the targets of the drugs that we develop for therapy, but also because, of course, going from RNA to proteins, there is a lot of regulation that goes in between, and then proteins can have different level of translation compared to the level of RNA. They can be modified, they can have different interactors, different localization, and so on and so forth. One silly example is that one of the most abundant proteins in the blood is serum albumin, but it's mostly not produced by blood cells, it's mostly produced by the liver. So you see that just looking at the RNA level of the individual cell types doesn't give you always the exact feeling of what kind of proteins are in a certain tissue, especially of a complex organism. So we need to do single-cell proteomics. Right? There are two big problems, though, in doing single-cell proteomics, and we sort of hinted at it already, and that is the fact that it's more complicated to amplify the signal of proteins compared to RNA and DNA, for which we have PCR, and it's much more complicated to barcode and multiplex cells when you do proteomic experiments, and therefore the number of cells you can analyze per experiment is smaller. In other words, with the current proteomic workflows that we have, it's very complicated to analyze 10 picogram of material, which is more or less the amount of material, the amount of proteins that is in a single cell. And of course, if you assume, let's say, that you're doing mass spectrometry experiments of a regular proteome, even if you run, let's say, 1 hour per sample, you need months to analyze just a few thousand cells. That's why when you look at single cell proteomic data, you see that the number of data points is always much smaller. Okay, there are solutions to all of this. Now I'm going to go through a little bit of histories to bring all of us up to speed on why we've gotten actually so far. So I'm going to start, of course, not going too much back, but going to what we think is basically kind of the foundation of the modern single-cell proteomics. And of course, we need to give a shout out to one of the main scientists who has been working on this technology, which is Dr. Slavov at Northeastern. And the idea, the first modern single-cell proteomic paper, let's say, the idea started from the fact that, okay, we don't have enough sensitivity to analyze the proteome of a single cell, but what if we use an isobaric tagging technique, like, for example, TMT, and I'll explain in a few slides a little more how it works, and we actually mix it with a carrier, so another channel where there is more signal to boost the signal of the proteins in the single cell, can we now detect proteins? And the answer was yes. And the answer was actually that they could actually perform the first, of its kind, single cell proteome using mass spectrometry. How they did it, we'll see it in a few slides. But it actually not only about how you tag proteins, there is an entire workflow that you have to consider. And there are, of course, differences compared to a regular proteomic workflow. So I divided it in these 5 sections that goes from cell isolation to data analysis. And there are, of course, differences in all of these steps compared to a regular proteomic workflow. And we're gonna look at them together right now. So first of all, cell isolation. So cell isolation, basically you need to deposit one cell in a specific well or in a specific plate, somewhere where you can basically prepare them individually. How do you do it? There are essentially 3 kinds of technique. You can use classical FACS sorting. The most modern one are actually able to deposit cells one by one, for example, in a 96 well plate or 384 well plate. And then actually you can do it, just like that. Method number 2, you use basically essentially robots. So liquid handlers that are able to work with very, very small volumes. And actually there are a number of handlers that are actually available on the market now that you can use. Some of them are very expensive, some of them are less expensive, but of course, the more expensive they are, the more features they can do, and we're gonna see that in a second. And then finally, for example, if you work with tissues, you can also consider about isolating individual cells by literally cutting them with the laser on the surface of a slide. And this one is actually a technique that is growing as we speak. Like it's very new and is starting to give the first exciting results. Okay, so speaking of liquid handler, you may have heard this before, but basically the most popular one is a device called Cell-N-One, from a company called CNION. The way it works is that it's essentially a FACS sorting machine, but in addition, it has a camera, and actually that's one of the most expensive part of the instrument, that is able to detect if a single cell has been isolated on their probe. So then when it gets deposited on a plate or a slide, you know that that single cell is present in that liquid droplet. And the way it works, there's a little video here. So what you see here in orange, you see the probe that goes around. This one right here on the left, this one was the camera. And actually what you see right now, you don't see a single cell being deposited. You see basically the probe mixing some cells and picking them up. But the concept is the same. There is this probe that goes around the deposit cells in this specific example on a slide. And now in this specific example, once it picks them up, it deposits them in a 384 well plate. So it's basically a liquid handler that works with very, very small volumes. Is one of the more popular approaches that we have today is, and I'm going to go into details on this method, is basically to deposit individual cells on a glass slide, literally like those slides that you use from microscopy. And then you see that each one of these spots is a single cell. And the reason because they are grouped this way is because we can barcode them and mix them and then analyze them together in a mass spectrometer. And I'll show you how. This one is some figure that actually basically show how these little droplets are delivered by this probe. And here you see basically a zoom in of an individual cell that is deposited on the slide. It's not the only method that you can use. There are other scientists that have invested more time on liquid handler that are objectively cheaper, and they actually essentially work almost like a printer. This is an example of the Uno from Tican, which was literally originally developed by HP, the company that makes printers. And the way it works is that there is a chip, and from that chip, literally, like cells are delivered, are literally spit one by one into a plate. It works well, its results are, really promising. The limitation of this, is significantly cheaper, but the limitation is that, there is no camera. It's a little bit more complicated to check if, what is the efficiency of the, of the deposition. But, good results nonetheless. And then regarding the laser microdissection, like I briefly mentioned before, this is technology that is emerging as we speak. And interestingly, it actually starts to use quite a bit of AI, right? Because once you take a picture with a microscope of a tissue, this is a piece of a liver, and then you want to basically tell the robot, please cut individual cells. The software needs to be trained with what is an individual cell. So basically what you see right here, you see a cartoon that basically tells you how the software is trained to recognize the border and the shapes of the individual cells. So then when it goes on and cuts them with this laser, it cuts exactly cells, one by one. The advantage of doing it on a tissue that is still like on a tissue format that is not being homogenized and dissolved into single cell suspensions is, of course, that now you can actually retain the information of the spatial position of these cells, and it can actually bring very interesting new dimension in the analysis. For example, in this specific example, the group of Matthias Mann studies how far away certain cells are from certain critical veins of the liver and how it changes their biology. Okay, now let's say that we have our individual cells on a slide, on a plate, and now we need to digest the proteome in order to be able to analyze it, like for a regular proteomic experiment. Now, here it starts the problem of having to deal with very, very small volume, right? Because we're working with nanoliters. Actually, mostly cells are dispensed into volumes that are around 8 nanoliters. So it's a extremely tiny volume. It's very hard to pipette manually. So how do we break these cells so we can digest them with trypsin or other enzymes? There are usually two methods that people use. One down here, the freeze heat lysis, so basically freezing and heating the slide or the plate. This is something that, to be honest, is not used that often, but people have been testing it and it gives some kind of results. The one that is actually used more often is to use gentle buffers that contain a certain percentage of detergent, but that percentage is relatively low because there is no really way to remove the detergent afterwards, after the sample preparation is completed, before you do the analysis. So usually those detergents are not very concentrated. But these methods, they work. There is ample space for improvement, right? So certain proteins are not easy to maintain in solution in such a tiny volume, also because this volume evaporates. So if one venue of research for single cell proteomics is definitely to improve the sample preparation. And then, of course, because it's complicated to add a lot of reagents, usually the step where we add the DTT and iodocetamide, when we open the disulfide bonds and then we alkylate them, that is usually bypassed. So you're only dealing with proteins that digest with trypsin in their natural state. Okay, so now we have individual cells that have been broken and the proteins have been extracted and they've been digested. Now, before we inject them into the mass spectrometer for the analysis, we need to find a way to multiplex them, right? Otherwise, we need to analyze them one by one. And so here it comes the usual story about what kind of quantification technology we use to perform this proteomic experiment. And I usually like to display this graph. It's quite old at this point, but it's still my favorite. That gives the idea about the different kind of proteomic workflows for quantification that goes from metabolic labeling, where you label cells or tissues like even before you start the sample preparation, to the label-free technology, which we all know, where basically things are injected in mass spectrometer separately before the analysis. Every method has its own advantages, right? So label-free is very straightforward. Using spike-ins has its advantages, and sometimes actually it's done in single-cell proteomics. For example, spiking in a very, very small concentration of proteins from a different organism, for example, E. coli, which can be used for normalization and for quality control because certain proteins need to be detectable. Chemical labeling are the most common, especially isobaric labels. We'll discuss it in a minute, which is also how single-cell proteomics started in 2018. And metabolic labeling is also used. To be honest, the classic SILAC approach, where you have condition A that is light and condition B that is heavy, is not used very often in single-cell proteomics. Now, to be honest, probably not a lot in general proteomics either, but it has one very big advantage and provides very interesting information when it's used in a pulse mode. Basically, when you actually grow cells for a certain amount of time on heavy amino acid media, so you can actually study how quickly proteins are being synthesized. I'm going to have a slide on that too. Okay, first of all, let me say one thing. When you do a label-free proteomic experiment, actually it can be very quick. So this one is a paper that is not published yet, but is present online as a preprint, and is actually really intriguing because the group of Ryan Kelly figured out a way to make a single-cell proteomic analysis by doing 5 minutes injections. So 5 minutes injection that requires complex chromatography setup. Then while you are loading one sample, the other one is being eluted and vice versa. But it actually seems to work well. And basically by doing 5 minutes injection, you can analyze quite a few hundred cells per day. It's a bit of a complex setup, but I'm actually looking forward to see how it develops because objectively speaking, label-free proteomics is one of the easiest approaches. So it really helps democratizing the technique. Metabolic labeling, like I said, is not used that much anymore to just compare sample A with sample B by doing different labels, but is actually used to study protein turnover rates. So you basically grow your cells in light media, then you transfer them in a heavy media, usually with heavy lysine and arginine. The new proteins, they start to be produced using heavy labeled amino acids, which basically means that now every protein will have two signals in the mass spec, and the ratio between the heavy signal and the light signal basically tells you how quickly that protein is being synthesized, right? Because a protein that is synthesized very quickly will have a heavy signal that goes up, in a very short time. And so basically, by doing an experiment like this, and the group of Jesper Olsen has been basically pioneering that, you can actually perform not only proteomic at the single cell level, but protein turnover analysis at the single cell level. And if you check out this paper, actually you'll see really interesting results. So it is possible. But let's go back to the techniques that are used a little bit more often, which is chemical labeling. Chemical labeling basically means that you extract your protein, you digest them, and then you add the chemical to tag these individual cells before you mix them together. There are two types of chemical labeling. One of them is isotopic labeling, where this creates a difference at the precursor mass level. So the tag that you add to proteins in cell 1, cell 2, and cell 3 basically leads these peptides to have a different mass. And this one is actually convenient because then you don't need to do MSMS, you don't need to break those peptides to tell from which sample they come from. The disadvantage is that they're usually very limited to just a few chemical labels, right? So you can, the common ones are 3, 5 different kind of labels. Is not that much. But they have this advantage that at least you divide your number of injection by 3 or by 5, and it can make actually a big difference, for example, between 1,000 cells and 5,000 cells that you can analyze. And the other advantage is that because the precursor masses have a different mass depending on which sample they come from, you can acquire data using DIA. So you can basically do this sliding window in the instrument where you fragment everything that goes through. And we're not going to discuss this because this is not the topic. But DIA compared to the classic data-dependent acquisition approach in mass spectrometry usually provides a bit more unbiased quantification and a little bit more reproducible quantification run by run. So doing actually DIA has its own convenience. It cannot be done with other type of chemical labels. We'll discuss it in a second. But actually, people have been testing it for single-cell proteomics with quite interesting results. Actually, this graph, this one is a PCA that is really impressive because basically what it's showing is showing that this group, again, Nikolai Slavov, they analyzed three different kind of cells. They analyze them not at the single cell level, so in bulk, and then they analyze them with two different type of mass spectrometers. And when they combine the data, they actually show that they can still group cell types together, despite the fact that they use different amounts of material and different kind of instruments, which we all know, what kind of big batch effects we have when we actually use different instrumentation to analyze the same type of sample. So these are really impressive results. And this again basically shows how DIA is really producing like very reproducible quantification, actually objectively more reproducible than with the classic data-dependent acquisition. But data-dependent acquisition still has like a role to play, especially in single-cell proteomics, because it's the acquisition method of choice still today for isobaric labeling. Isobaric labeling, what I mean by that is the family of chemical tags that includes, for example, TMT and iTrack. iTrack was developed actually now a long time ago, and the idea of these chemical labels is that you basically tag the proteins from different cells with a molecule that has an intact mass that is shared in all of its configuration. But then what is unique about them is that once you break this peptide and you perform MS-MS fragmentation, there is a piece of this molecule that falls off. We call it reporter ion. And this one has unique masses that we can use to tell apart the individual cells. How does it work? Simply because the distribution of the heavy atoms, so carbon-13, nitrogen-15, deuterons, are in different areas of the molecule. So when you break the molecule right here, this reporter ion will have a certain mass. When you break it right here, this reporter ion will have a different mass. But at the end, these molecules, they all have the same mass at the precursor mass level. This is a little bit more of a overview about how it works. And actually, this also tells you why it becomes very interesting to have a carrier channel. I'm going to tell you in a couple of slides how that works. Because using TMT is exactly how modern single-cell proteomics started, like in 2018. The other advantage of using isobaric tags is that now different companies are becoming more and more skilled in designing tags that are more and more complex in terms of how many combinations of heavy isotopes you can have in different regions of the molecule. So now there are commercial kits, for example, TMT, that can go up to 32-plex. So we're talking about tenfold more than what you can do with the isotopic tagging. And that actually shrinks a lot the number of injections that are needed to produce a population of 4,000 or 5,000 cells. And like I said, this was exactly how it started, right? So because all of these tags, they all lead to the same intact mass, basically what happens is that at the end, when you mix multiple cells into the same injection, you have a signal that becomes as intense as the sum of all of these cells, which means that if you have a channel where you add a significantly more amount of material, you will actually significantly increase the intensity of your precursor signal that becomes more detectable by the mass spec. It can more easily select it and fragment it. This is actually how SCOPE-MS actually started in 2018. And then basically, the authors realized that, okay, one of the problems that we have in mixing 200 cells with individual cells is that this 200 cell channel becomes a little bit, off scale when we need to normalize our individual cell signals by this reference. So they come up with a new technology a year later, where they call it SCOPE2, where basically now they introduce a second channels. So basically, let me show you this, how it works in the next slide, and it's more intuitive. Now you have, let's say, 32 channels. Back then there were fewer, but let's say now we have 32 channels. 30 of them, they correspond to reporter ions to each individual cell. So you mix 30 different cells into the same atom of tube. Then you have an extra channel where you put together the signal of 5 cells, and this one is going to be the reference in every injection. So these 30 cells are going to change cell by cell, sample by sample, but this reference will always remain the same. So in the end, you always have ratios to the reference. In the end, all of your injections are comparable because they've all been normalized by the same reference. In addition, there is another channel where the carrier now is present at a much higher concentration. This one is not very good to use to normalize individual cells because it is, the dynamic range is too wide. But this one is actually used to boost the signal of the precursor ion, and that's how it makes individual cells more detectable. It works well. That's basically the technique that is still at the moment the most popular today. Then one of the problems that comes up is that, okay, if we acquire spectrum using this, this type of labeling method, then we need to do it with DDA, right, with data-dependent acquisition. So now the problem of data-dependent acquisition, as you all know, that is a little bit stochastic, right? So because a different injection, you could actually end up selecting different signals, and especially when the signal is very, very low, like when you do single-cell proteomics, it's quite probable that you're going to have stochasticity, so that in different injections you're going to select for fragmentation different peptides, so you will end up with a proteomic table with a lot of missing values. So the third iteration of this method, scope 3 or P scopes, that stay for prioritize. Now the authors, they introduce max quant live. So basically, as the sample is running in the mass spectrometer, what happens is that the instrument does not unbiasedly select whatever is going through, but it tends to prioritize signals that we decide are the most relevant. How do you know which ones are the most relevant? Usually you do a previous injection with more amount of material, so where you can produce this sort of semi-targeted list. And so then when you run the single cell samples, the software knows which ones should be selected first. So you actually reduce significantly the number of missing values per injection, and you basically end up with a table with much fewer missing values that here are depicted with the white sign. So 3 iterations of this workflow they took, but now we actually have a workflow where you can inject more than 30 cells at the time, so you can actually improve quite a bit the throughput of the analysis, and you reduce a little bit, actually quite significantly, the number of missing values in the final table. Where do we go next? The way we go next is potentially combining isobaric and isotopic label. Like, so if you actually can have different kind of labels on one side of the peptides, another label on the C-terminus of these peptides, you can actually multiply the number of different labels and the number of cells that you can mix together in the same injection. And this is just an example of how many cells you can analyze in a day if you actually come up with a good system to chemically label properly your peptides. It's complicated, right? Because as you know, chemical tags are usually labeled 3 amines, so which is the N-terminus, and usually the lysine at the side chain of the lysine at the end of the peptide after lyse digestion. So it's a complicated chemistry, but that is actually what's coming next. Okay, so now we discussed about how to isolate cells, how to break them, how to label them, and how to inject them in the mass spectrometer with what kind of acquisition methods. Now, a couple of slides on what kind of mass spectrometry is being used now to do single cell proteomics, and this one is one of the more rapidly growing markets in in the field. Those of you who go to ASMS, they know what I'm talking about. So there are instruments that come out every year with a little bit more sensitivity, with a little bit more speed, and now they come basically to have instruments that are designed to be very efficient to do single cell proteomics. The leading company is not a secret, are Bruker and Thermo Scientific. And then, there are certain approaches, even in liquid chromatography, that are being optimized on purpose for single cell proteomics. So columns that are designed for single cell proteomics, and also chromatographic separation with certain flow rates that are designed for single cell proteomics. So it's a market that is growing very rapidly, and of course, the labs that can afford it, they take the latest technology, and actually now we can get really surprising results. So I didn't want to miss the opportunity also to say a big shout out to the lab of Karl Mechtler, which is also a lab that actually spent a lot of time optimizing single cell proteomics. And you can see that now with the most modern mass spectrometry, you can detect thousands of proteins per single cell, which is actually an unprecedented type of sensitivity, really, really impressive, which basically means that we are not too far from doing global proteomics from single cell. So the technology is about to be ready. The methods are about to be ready. There are a series of optimizations that can be done, as I told you along the presentation. But single cell proteomics is a reality. Okay, so now it comes maybe the most, I hope, the most interesting part of the presentation is that, okay, let's say that we can perform a successful experiment. What do we do with this kind of data, right? So what kind of information we extract that we don't have from regular proteomic experiments? So first of all, before we get into how do we interpret the results, there are a few things that are important to consider, right? So single cell proteomics remains today a challenging type of experiment, and the success rate of the individual injection is of course not as high as regular proteomics. So of course, quality control is very important, right? So because compared to regular proteomics, we're going to have more missing values, we're going to have more significant batch effects, variability, because the signals are very low. And then, of course, depending on what kind of cells you use, you can have very different results. Like, so stem cells are very small, so they're going to give you single cell proteins with much, much shorter list compared to hepatocytes that are much larger type of cells. So also, you can note that when you look at these publications where people show like a lot of proteins identified from single cells, they always do it with cells that are, quite fatty of protein, very rich of protein content, right? So obviously, when you start studying like glial cells, stem cells, T cells, immune cells, cells are significantly smaller, so things are more challenging. So don't despair if your list don't look anything like Karl Mechtler's lab, for example. Okay, so this one is a typical workflow on how you filter data before you get to final results when you have a single cell proteomic data set. We're not going to go through each individual point, it's not important, but just to let you know, there are some steps that are unique when you do single cell proteomics. And then, of course, this is important to consider. So whenever you use one of the existing scripts or you do your own data analysis, take into account that it's not the same exact type of analysis that you do with regular proteomics. For example, one thing that is important to consider, like I said before, when you inject single cells, whether they are multiplex or not, whether you're doing chemical labeling or label-free, you can get, injections that are successful and injections that are very not successful. Right? So this one is an example of number of peptides that are detected per injection in the mass spectrometer studying single cells. And you see that there are instances where the number of detected peptides is much, much less compared to others. So these are obviously runs that if they are kept into the analysis, they're going to skew a lot the statistics and the biological interpretation. You can see them also displayed in a PCA plot, how they cluster far off, compared to the other. So we actually, in most of the workflow, the recommendation is to actually eliminate those runs. That will lead you to final results with fewer cells that you can display in these graphs, but it's still better than keeping in consideration as a true biological variation, something that biological variation is not. This is clearly technical variation because it is not really feasible that one cell has 5,000 cells, 5,000 peptides, another cell has 600. So clearly something went wrong with the injection. If you keep this kind of injection, you're basically introducing artificial variation, which at this point is very important at the single cell level because the variation of the proteome is exactly what we are looking for. So don't keep into your pipeline injection that clearly have technical problems. And this one is an example of the type of batch effects that you can have when you do single cell experiments. So, for example, this one was an experiment where they've been injected like many injections, and what you see the letter here, this is the position of the sample in the plate. So it was injected, one after the other, and you can see when you display the results, the proteomes as a PCA plot, you can see that you get clusters that are basically clustering based on the injection type. So those that were injected late, they cluster all here on the left, and those that were injected early, they cluster here on the right. So this is actually very important to visualize. So make sure that when you run your quality control, you always have graphs that actually take into account this aspect. Like, so do I have a batch effect, right? And specifically here, this was an example where 3 columns have been changed during the analysis because they got clogged and they needed to be replaced. And you see that once you introduce a new column in the analysis, you get proteomes that are slightly different from the other. That's a problem. And so you need to go through some batch correction, which I will discuss in a couple of minutes. Okay, so I'm not too far from the end, but obviously I want to spend a little bit of time telling you about the data interpretation, because this is actually the most important part when you do a single cell proteomic experiment. Why do you do it? Why do you go through the trouble of making a challenging experiment with producing data that are different from a regular proteomic experiment? And the main point is that you want to be able to tell what is heterogeneous in your cell population, right? So here, for example, this one is an experiment that we did actually in our own lab, where we have liver cells from a mouse that is 2 months old versus a mouse that is 22 months old. Like we discussed before, of course, aging makes the proteome of the cell different. And in fact, when you plot this proteomic data in a PCA, you can see that you can separately cluster cells that of the mouse that is young from the cells of the mouse that is old. And obviously, these cells, they're all mixed together, with the TMT labeling in the same injection. It's just we know which cell is which, so that's why we can color the graph. But it's not that these were injected first and these were injected later. They were injected all together, intermixed. So one information, of course, like I told you, you can separate young and old. Now, the next question is, are the old cells more heterogeneous than the young cell? Now, from here is a little bit difficult to tell, but actually, they tend to be, especially when you look at cells that are very old, from organisms that are very old, they start to be actually different from one another, not just from the young cell population. And then the other information is, okay, if we focus, let's say, within a certain group of, within a certain phenotype, so within the young cells, for example, the cells that are separate in the graph from each other, is it because they are different cell type? So if you're working with the tissue, very likely they could be. So what you see here, for example, you see a type of representation that you cannot do when you do regular proteomic experiments. Basically, you can display your cells on a PCA plot, of course, separated by the proteome, and then color those cells depending on certain biomarkers. For example, here you can see that you have a separation between the proteomes of cells that are close to pericentral veins in the liver and cells that are close to the periportal vein in the liver. So it's the same type of graph, colored differently depending on certain biomarkers. You can see that you can actually start to separate cell types within the same population. And this is actually very exciting. This is something you cannot do with regular proteomics, but actually there is more, because if you acquire if you collect and prepare your data, for example, using a device like the cellular one that has a camera and therefore can give you some phenotypic information about your cell, for example, the morphology, the shape, the size, you can add additional dimension further. For example, old cells in a liver, they tend to be a little bit more fat. So they tend to be a little bit bigger compared to young cells. They have a similar protein content, but because they accumulate more fat with time, they tend to be, a little bit bloated compared to the young cell. So you can even separate cells by proteome and by morphology. And you can do this at the individual cell level. And now imagine if you're studying, for example, an immune cell population, like how many dimensions of data you can deconvolute from a single experiment, right? So what kind of proteomes, what kind of subclasses, what kind of conditions, what kind of morphology, and what kind of biomarker. So now you're talking about 5, 6 dimensions, things that you cannot do when you look at global proteomics. Does it go further? Yes, it goes further. And now you can take inspiration also from existing publications, reviews, where people discuss about what is the potential of having a single cell proteomic data set. And this one is one of my favorites, where you see that there is a discussion in this review where people discuss that the authors discussed that beside grouping cells based on cell type, which is one of the most typical applications, you can actually, for example, group cells according to their cell cycle, or according to the time of infection, or according to how long they've been subjected to a drug treatment. And then basically, even if these are not different time points, it's the same cell population, you can actually group them by pseudo time. So it's very clear that this cell has been in contact with the drug more than another. How is the proteome changing? When you look at covariation, you can even study what kind of proteins are regulated together, like in individual cells. And now that you have many, many, many cells, you can actually get very good statistics and, for example, start to predict new complexes, right? Because a protein that is always correlated with another in thousands of cells, they probably have some kind of interaction. And then, of course, the next step would be to integrate protein data with RNA data, for example, to study changes at individual cells of, for example, post-transcriptional regulation and translation, which is something that is coming very soon. And this one is actually one last slide on this aspect. Now we have the contribution also of the lab of Erwin Schoff in Denmark, that is also working very hard to generate very interesting single cell proteomic papers. Basically, now here they show how by integrating protein and RNA data, you can clearly show that proteomic data, they actually do a better job at resolving the cell state of this cell population compared to using RNA. So that's another shout out to not give up on single cell proteomics, even though it's significantly more challenging than sequencing, because it has major advantages. Is it as complicated as it gets? No, I want to spend one last minute telling you about protein modifications, right? So that's another dimension of the proteome that makes it more complex than transcriptome. And this one, actually, I want to dedicate it to my lab, because people have done, an amazing work, developing techniques to study protein modification at the single cell level. Especially have some lab members that are working very hard on single cell methods, while others are focused on other things. And this one is an example of a workflow where we basically implemented single cell histone post-translational analysis, right? So these are the results. It's actually kind of amazing how, histones are relatively abundant proteins, so they are a little bit easier than others to detect, but it's kind of amazing how at the single cell level, you can see chromatograms of modified peptides that are really crystal clear, nicely distinguished from the background noise. This one is an original paper where it's been shown that you can study 60, 70 type of different modification per cell. And why this is relevant? This is relevant because now, compared to the technical variability, now you can study the biological variability of protein modification. And the reason because this is important is because now you can see that when you look at things cell by cell, you can see that there are certain type of modifications that are very noisy cell by cell, and they can basically describe region of the chromatin, so of the DNA compaction, that is more unstable compared to other instead that have a very low biological variation cell by cell. You can even like compare those variations, like I said before, and doing basically co-regulation analysis. And what is interesting here is that in certain conditions, certain histone modification, they change in abundance at the same rate between one cell, in the same cell. And this one is actually very exciting because you can basically study crosstalk between post-translational modifications. Say, okay, if one modification is present, the other one needs to be present too. Therefore, they are either catalyzed by the same enzyme or the two enzymes, they communicate somehow. And why do I spend time telling you about this last thing? Is because it gives me a chance of telling you about validation experiments. Now, so you produce this very complicated type of data. Do we have now orthogonal methods to prove that this data is solid? And for example, you can use actually FAC sorting now, because you can actually have antibodies, including produced by Protein Tech, where you can actually label the same cell with the two markers that you think are co-varying in the same cell. And here you can show that indeed, if you do the experiment with antibody, when one histone modification is more abundant, the other one is more abundant too. So we sort of validated the crosstalk between two different modifications. That's how I wanted to conclude. And I think it's perfect timing. So just one last conclusion slide. I hope it was interesting. I hope you guys, had fun. And indeed, single cell proteomics is now a mature technology. Like, it's challenging, but there are a number of labs that do it. Actually, they are starting to emerge also labs that do it as a service. So in case you don't have the means to do single cell proteomics in your own lab, still, I hope this presentation was important, because you can start to think about what kind of information I can extract from these experiments, which go beyond regular proteomic experiments. We discuss what kind of workflows we use. We discuss what kind of bottlenecks there are. We discuss about why it's so important to study proteins, not just the RNA, even though single-cell transcriptomic experiments are objectively a little bit more straightforward. So with that, I think I'll stop here. Big thank you to the lab, to all of you for listening, to the funding, and of course to Protein Tech to sponsor this really cool series of seminars. I hope we will see each other once again.
Great. Thank you so much, Simone. You can see in the comments we've gotten a lot of appreciation for your talk. I think a lot of people learned quite a bit. So before we begin the Q&A session, I do just want to share a few more slides, and then we will get to some questions. Alrighty. All right. So I just want to highlight a few products that might be interesting for our audience today. So I did see a couple questions asking about how to study the transcriptome and proteome simultaneously. And so we do provide a solution for that, which is our line of multi-pro DNA barcoded antibodies from Protein Tech Genomics. So we offer several multi-pro products, including our human discovery panel, which is compatible with 10x Genomics Flex. It is the largest and most thoroughly validated DNA barcoded antibody panel for concurrently detecting cell surface and intracellular proteins and gene expression at single cell resolution. The Discovery Panel provides deep insights while providing twice the antibodies than others on the market while costing less than half. We also have our HashMax cell hashing DNA barcoded antibodies, which enable higher sample multiplexing and improve data quality from your single cell RNA-seq experiments. HashMax antibodies work by providing samples with unique DNA barcodes, which enable maximal sample loading. Additionally, these antibodies allow for robust doublet detection and removal, which enhances your data quality. And then finally, we also have an upcoming product called the Spatial Explorer Panel, which is the first high-plex protein detection solution available for the Visium HD FFPE. So this panel contains 200 antibodies against diverse protein targets. The panel has been extensively tested on diverse FFPE tissues and pre-titrated for ease of use. Early access for this product is available, so if you are interested, please feel free to reach out. I also wanted to highlight a couple of new kits that we've recently launched to assist in oligoconjugation. So the first is our flexible oligo-ready antibody labeling kit. So if you're familiar with our flexible technology that uses a high affinity linker to quickly conjugate primary antibodies, this is the same concept, but now we've extended it to conjugate antibodies with oligos. So this kit works in two phases: the initial conjugation of your oligos to high affinity FC specific flex linkers, and then the actual labeling of the primary antibody with the oligoflex linker complex. So depending on your needs, each flex linker will carry one or two azide groups, which react with DBCO oligos through click chemistry. After the conjugation, the protocol also has a step to remove any excess free oligo to prevent background signal, and the recovery rate of the conjugated oligo is between 60 to 80%, so quite a bit higher than traditional methods. Each kit also allows to prepare up to 3 different oligoflex linker complexes, so you can create your own custom barcode library, which can be stored long-term at -20. So once you have your oligoflex linker ready, you can then label your primary antibody. And what's great about flexible is that it truly is flexible because you can label anywhere from zero point 0.5 micrograms to 100 micrograms of antibody, depending on what you need. And the labeling is actually compatible with a wide range of antibody buffers. So you don't have to worry about doing any sort of buffer exchange. The protocol for the antibody labeling is also very easy and it just takes 15 minutes. So you simply mix your antibody with your oligoflex linker and some flex buffer, wait 5 minutes, add in the flex quencher, and then that's going to bind to any of the unbound flex linker, wait 5 minutes, and then your antibody is ready to use. And because you are pre-labeling your antibodies, you can multiplex quite easily, even when using same species antibodies. If you are using a conventional method for oligoconjugation and are set on this method for your experimental needs, we also recently launched our CleanApp kits that can help with a quick and gentle method to remove excess free oligo from your sample. So the CleanApp beads in the kit bind directly to your oligoconjugated antibody, which can then be washed and eluted to obtain a pure product without free oligos. The kit is compatible with either post-conjugation cleanup or after on-bead conjugation. If you are familiar with oligoconjugation, you might know that a common approach to removing excess oligo is with protein A. However, our CleanAb offers several advantages over protein A method. So first, CleanAb shows superior binding performance to IgGs. The CleanAb beads have a very high affinity for either rabbit or mouse IgG, and you can see that with the Kumasi stain here on the right, where there is virtually no conjugated IgG in the flow-through fraction when incubating with the cleanup beads, whereas there is some still left over after incubating with protein A. Secondly, the cleanup kits offer gentle elution of the oligoconjugated antibody at a native pH of 7.5, unlike protein A, which requires an acidic elution. This native elution is very important to preserve protein conformation and biological activity of your antibody. And finally, the clean ab kits allow for high recovery of 60 to 90% of your product compared to protein A, which can frequently have incomplete elution. So you lose quite a bit of your product because it's still going to be stuck on your beads. So if you are interested in trying out this kit, we are offering a free sample for the attendees of today's webinar. If you'd like to request a free sample, you can do so by filling out the form after scanning the QR code that's going to be on right over here and then also on the last slide. So you can scan that QR code and fill out the form. We'll also send a link to request the sample in a follow-up email. And then lastly, I just want to highlight real quick that within our expansive portfolio of antibodies, some are offered with different buffer formulations, including PBS only. So our PBS only recombinant antibodies are developed using our proprietary ABCE platform, where we can monitor and select clones for optimal expression and performance. All our PBS only antibodies are carrier-free with no BSA, no sodium azide, and no glycerol. Because they are recombinant, they come with the advantages of excellent specificity and high affinity, high purity, and lot-to-lot consistency. And with no additives, they are suitable for various downstream applications, including conjugation and mass cytometry. So with that, I just want to remind everyone that as an attendee of today's webinar, you will receive two follow-up emails. One will have a recording of this webinar as well as a request form for your free sample of our Clean App Kit. And then you'll also get a certificate of attendance. And we do ask to please allow 1 week for these follow-up emails to be sent. So with that, we can go ahead and begin the Q&A. If after the Q&A you do have any further questions, you can always email us at events@ptglab.com, or you can reach out to Simone directly, and I put his email address right there. And I'll pop that slide back up in at the very end again. So all right, let's go ahead and start the Q&A. I know we're a little over, so we'll just cover a few questions that I had jotted down. So the first question is asking, why do we need single-cell proteomics when the tissue as a whole is affected in disease? And having single-cell proteomics data can just add noise because we are not going to treat a disease on at a single cell level, whereas targeting a tissue.
Yeah, I like that question. Actually, I know this is the most thought-provoking, like in the pool, and I'm happy that it came up because the person who asked this question is right. Like, so single-cell proteomics creates noisy data, and the question is the noise relevant, right, in the biological system? And we might argue that actually it's very important, right? So I agree. Like, we don't cure a disease by treating one cell that is different from all others, but by knowing how cells are different within a population, allows us to understand why certain drug therapy works on certain cells and not others. And also, it could also, now, for example, if we think about cancer, there is a big talking about cancer stem cells. So what are the cells that actually generate cancer cells and then differentiate and then become uncontrollable? What is unique about those cells is very critical, but we don't know it yet. And therefore, like, understanding in a cell population, if there are unique patches of cell types that have unique proteomes, and those unique proteomes, they suggest that these cells are primed to be uncontrollably proliferating, for example, is a very important piece of information. But I don't want to make this too long, but the point is, I would conclude that this is a very good question because for a lot of this noise, we still don't know how to analyze it. And therefore, we're all needed, right? So we need not only the classic approach to data analysis, not even ChatGPT can give us this kind of answer at the moment. It has to be new ways of looking at data and understanding why the noise is relevant.
Great, thanks. The second question is asking, so the turnover time for proteins is very different from that of RNA. Does this have to be considered when we are doing single-cell proteomics, especially if it is expensive?
Yeah, that's true. Yeah, no, actually, we're only scratching the surface at the moment on, why protein turnover is important in biology. So the answer is that, of course, it is, right? Because certain proteins are very stable in a cell, and certain proteins are synthesized at a very fast rate. And if you think, for example, at the drug that creates a covalent bond to a protein, if that drug targets a protein with a very fast turnover rate, it gets eliminated very quickly, right? Because there's constantly new protein that is being produced, and the drug gets diluted away. So yes, the turnover rate is extremely important. I like to use this metaphor. When you enter a household and you want to know, what's happening in that home. Normally, you don't look at the furniture because those are the same every single day. You look at the trash bin, right? Because from the trash bin, you can see, what people have eaten the day before, if there was a big fight and some plates were broken. So looking at the turnover of proteins rather than, just what proteins exist tells you quite a lot on the dynamic of a biological system. At the single cell level, it's still very complicated to do a comprehensive protein turnover proteomics, but as I show in the presentation, it's coming, and I think it will reveal very interesting information.
Great, thanks. So the next question is saying, how do you decide the labeling method based on specific experimental design and application required? It seems label-free gives more depth, but isobaric gives better resolution. Will it hold true here as well?
Yes, and that's actually true also in regular proteomics. So it's nothing new for single cell. The big difference with single cell is that you want to analyze a lot of cells in order to have enough information on the heterogeneity of a cell population. So that's when isobaric label that can be multiplexed at very high numbers gives a big difference in terms of throughput. So if you were to perform, let's say, a canonical single cell experiment, I would recommend to use isobaric labels, the one that is used most often. Label free, I agree, gives more depth and is probably more unbiased in its own way. But at the moment, we'll see what Ryan Kelly develops. But at the moment, the throughput is a bit limited. And therefore, if you want to study a large population of cells, you need to have, a lot of instrument time available. If you have it, that's great. But most laboratories, they don't.
Great. I think we have time for maybe a couple more questions so we don't go too over. So the next question is, how do you deal with the isotope impurities of TMT? How impactful is the carrier on other channels slash single cells?
Yeah, it's a good question. Like I didn't go into this kind of details because they were very technical, but that's a very good point. So the carrier, which is the most intense signal in the mix, usually on its side has an empty channel because otherwise the isotopes of the carrier will go and contaminate the carrier signals from the single cell. That being said, for the other signals, when you buy a kit, usually it's written in the brochure, because it has been tested, the level of interference of the various isotopes to the following channels. And so you do the analysis exactly the same as you do a regular proteome. But it's true, with single cell proteomics, you have this unique problem, which is one of the signal is much, much higher than the others. And that's why we usually leave a blank in the following channel to avoid interference from the isotope.
Great. Thanks. So the last question here is asking, what criteria do you use to distinguish true zonation-driven proteomic patterns from noise in single-cell PCA space?
Hmm. Yeah, that we don't have a very good answer. It's a very good question. Like so, I would say those zonation experiments, the ideal scenario would be like I show in my presentation, being able to cut individual cells according to their zone and therefore have a positive control, like that. It's not always possible. The throughput is still very, very low. And of course, the sensitivity is not as good as when you actually isolate single cells that are in suspension. So the lab of Matthias Mann has shown that it can be done, and that's great. But in terms of, let's say, routine experiment is still challenging. So indeed, when you study a tissue, usually cells are suspended, and the position of the cell in the tissue is usually inferred by the protein you detect. If you have biomarkers, proteins that are very well known to characterize a certain zone compared to another, you can use it. How confident you can be with the type of information, that is something that we are exploring as we speak. And of course, the more input, the better.
Great. All right. Well, I know we didn't get to a vast majority of the questions, but if you do have any questions for Simone, like I mentioned, you can reach out to him directly. I'm sure he will not mind. So I am just going to put his contact information right here again. And if you have any kind of general questions, you could always reach out to us at events@ptglab.com. And if you liked today's webinar and you want to stay up to date with any upcoming webinars, please do follow our socials because we post about all our upcoming events there. So with that, I think we can go ahead and end the webinar. I just want to thank everyone for joining, and thank you, Simone, again for your wonderful talk and your insights. And I hope that everyone has a good rest of your morning, afternoon, or evening, depending on where you're tuning in from the world. Thanks, everyone.
Thank you.